shown that twins who have same genomic sequences, so identical twins, they still show different phenotypes. Why do they show different phenotypes? Because genome is not the sequence. Genome is also other proteins that bound again which regulates the epigenome. So epigenome poises or predisposes individuals for different diseases and disorders. So it helps in explaining the cause of disorder. So for example, twins one may have obese and one may not have obese. And recently it was shown in Cell that there is a epigenetic modifier of PRIM28 which modifies the epigenome in such a way that one of the twins gets obesity and another not. So, and it was published in Cell by Delgado et al. So when we study epigenome, we also study epigenetic modifiers. So the field of epigenome also includes epigenetic modifiers. And then in plant genomics, epigenome has a big promise that it can help in improvement in the crop quality or yield. For example, a simple experiment done on rice plant, we just play with one of the epigenetic modifiers, is a seed, and then we see a difference in the size of rice plants. So the work that I was showing was work of somebody else, but we have also done huge data analysis with epigenome and promising results. For example, we have done epigenome profiling of postmortem brain of people with autistic disorders. So we took samples from the brain, from 3 regions of the brain. So you can see prefrontal cortex, temporal cortex, and cerebellum of at least 45 autistic patients and 49 controlling groups. We didn't have autism. So we did epigenome profiling and we used histone modification called H3K27 stylation. And we found many differential peaks, almost like 5,000 plus differential peaks between patients and control. Doing the same study using gene expression, RNA-seq, we could not find that many number of differential genes. So what is happening is that noise comes in gene expression data. Because if a gene is transcribed to RNA, by the time you start profiling the RNA gets degraded. And RNA is also related by after transcription. So post-transcriptional modification and degradation of the RNA causes a lot of noise. That's why we cannot get so many differential genes in this study, but we got a lot of differential peaks because epigenome remains conserved because the role of the nucleosome is to preserve the DNA. So chromatin is more preserved than RNA itself. So that we got almost 5, and it was a surprise to the field. And we even correlated the differences with respect to the underlying sequences. And what is the biggest promise that we had from this study? It was published in Cell last year, just one month ago, is that autism is not just one disease, it's a spectrum. It's called autistic spectrum disorder. So sometimes people have autism and it's very hard to detect it because they show different phenotypes. Some have a problem in reading, some have a problem in socializing. And what we found that, so there are different kinds of autism, like idiopathic, or because of the duplication of a region in chromosome 15, the group 15Q, we found that the differential peaks from these different spectrums are almost same and they have the same direction. So, this is the big surprise with the field. Why? Because companies could not design the drug or people say, okay, it's very heterogeneous. We don't know which site to target but now, since we have already shown that at epigenome level, the difference is similar. So this opens an opportunity for drug design companies which can target the epigenetic modifiers for autism. So epigenome study holds a big promise. Yes, and this study of ours. So it seems that okay. So I'll give an overview. So I told you that okay, epigenome can do this and that, but how epigenome profiling is done? So basically, you might have heard about ChIP-seq. So in ChIP-seq, what we do chromatin immunoprecipitation. So DNA is bound by the nucleosome, and DNA is also bound by different proteins. And nucleosome itself have histone which have different modifications. So we first fragment the chromatin, and then we use antibody specific to the protein of interest. Pull the small fragments of DNA bound with the protein, and then we remove the proteins. We then isolate the DNA from the protein complexes. We get short fragments of DNA, and we put these short fragments of DNA into the machine. As I showed you that it goes through the bleach PCR process, and these machines then create small. Then they read the small fragments of the DNA and tells which fragment of DNA was present in how much amount. So if suppose this protein binds region A and it doesn't binds region B, then we will have more number of sequences, small strand from region A. So coming to understanding this, suppose we have protein bound here, and so these are the small fragments of DNA sequenced by the machine. What we do is that, so they can be in both directions, both the strands, because DNA is generally double stranded. So we take the aligned reads, we count how many reads actually are coming from this location. And this count becomes signal. And this is where we come into picture. We do signal processing. And genome has a lot of biases. Because when we think that our genome is just one unique sequence, it's not like that. The sequences get duplicated in our genome. That's called copy number variation. And then while performing these experiments, in PCR, we have so much artifacts. So the noise percolates in this data. So processing of this data needs some expertise and experience. And that's why you are here. For example, if we say STK27 acylation data in mouse cell, and then in germ cell that we publish in developmental cell. So we use a protocol for small scale. So basically this data was generated using 5 million cells. This data was generated using only 100,000 cells. So we had to use some techniques to amplify the DNA, and that amplification created noise. We use random primers. So the lesser amount of sample we get, the challenge increases. Okay? So this is the problem of removing the noise and artifacts due to experiments or due to the genome itself. And that process we call it as finding in which site, or we call it learning tree crawling. Okay, so what are different kinds of epigenomic signals? Or we can say in big data, we can call it what is the dimension of the data. If you have a matrix, if you have genome as rows and columns as epigenomic markers. So I will talk about the dimension of this data that will be possible in one cell. Suppose if you take one cell type. So there are more than 130 histone modifications. And people have profile only for maximum 14 histone modifications. And that too not in all the cells, just for one or two cell types. So you can see more data is yet to come. And different kinds of histone modification can be classified into histone acetylation, methylation, proteinylation, ubiquitination. And they follow, there's a histone code. What will happen if acetylation will be there, and what will happen if methylation is there, and exactly which kind of histone acetylation. So that I'm going to talk about. And then we have transcription factors. Approximately 2,000 in human genome. And these transcription factors, they regulate, actually, the regulatory size, what we call as enhancers or promoters. Nothing but the play of transcription factors. And of course, histone modifications too.
Excuse me, sir. Can you explain protonylation?
What?
Protonylation. Protonylation. Third. Protonylation.
Yeah, I can explain to you. So you know acetylation. So when we add one more oxygen to to that acetylation, and because I can tell you you meet me after, I'll tell you the exact chemical formula for protonylation. It was discovered very recently, and it marks active region. So I don't remember the exact formula right now. So what we see is that proteins can bind different regions of DNA, and promoters, they come together as promoters through different regions. So basically they can bind away from the promoters, still the DNA can scaffold. And this forms a regulatory factory, a 3D regulatory factory. Okay? And then they control the gene expression. Based on the type of transcription factors bound and based on the enzymes, the transcription factor we put, the gene expression or repression is happening. And then there is open chromatin assay. So basically chromatin can be euchromatin or heterochromatin. Heterochromatin is when these nucleosomes are bound tightly, and euchromatin is when these nucleosomes are slightly open so that gene transcription or DNA replication can happen. And there are different assays to measure how much DNA is free or open. And like DNA-seq, ferri-seq, solo-seq, ATAC-seq, and MLA-seq. Okay, so we have shown it recently in our Nature Biotechnology paper that these different assays shows different kind of proteins. Like at enhancers, we have different kind of features from promoters. As at some places, nucleosome is completely evicted out, so the only transcription factor is fine. And this is also associated with the physical property of the DNA there, how much it is spent, and one can go in more detail. And in fact, MNAC can predict the nucleosome occupancy of the DNA. Okay, so
those others are the types of digestion protocol, or what are those others we have shown in the last slide? Sonication, DNA, so is it a digestion protocol?
no, these are different assays. It's not digestion. Digestion is done only in DNA-seq. Very sick, there is no digestion. It's just based on sonication, and it's a special way the way we sonicate the DNA. And so basically, we assume that the DNA which is loose, it will break more. And then we have sonoseq is also based on sonication. There's slight difference in the way we sonicate the enzymes that we use during sonication. So it's not only transcription factors is not the enzymes for digestion, it's just for the regulation. And histone modification is also involved in regulation.
So are there different types of sequencing?
What?
Are there different types of sequencing you are trying to say?
They are not frequently. There are different kinds of signal. Not even so. If you are talking in astronomical way, so you can say it's not frequency. It's just that
Sequencing, not frequency.
what?
I am trying to ask, is it you, what you mean is that these are different type of sequencing?
Sequencing? No,
Yeah.
these are not these are not different kind of sequencing, but these are different kinds of data. Like if you have 5 proteins A, B, C, and D, they bind in the DNA. So if you have genome-wide profile of these 5 different proteins, and you use the same sequencing technique to profile all that, so you get 5 different kinds of epigenomic data. So you have 5 chip-seq data for 5 different transcription factors. So it's not different ways of sequencing. So as I showed you in the earlier slide, like sequencing can be done using long single file sequencing, ligation sequencing. That is completely different. This is like the kind of information that we are getting from the chromatin. Then what proteins are binding here? What protein modifications are present in these 3 billion sequences? So we have 3 billion sequences of DNA, and proteins are binding. So this information says what kind of protein binds there and how much is the chromatin open there. Okay, so that's what epigenomic signal. It's not, and they can go through the same sequencing.
One more thing, sir. I'm a little confused. Like, epigenome, what I understood is the sequencing of the changes, modification in the nucleotide. It's not the change in nucleotide. So what I'm trying to see, whatever you state is like.
No. Epigenome is not the sequencing of the changes in nucleotides.
No, I'm saying modification in the nucleotide.
Modification is not nucleotides. Modification in the protein. Because I told you that nucleosomes consist of protein called histones. So their tails are hanging out. So it's the modification in the protein bound to the DNA.
It's the modification of histone protein actually.
Yeah.
In this case, suppose I am taking a X protein, for example, one kind of transcription factor, and doing the sequencing with different kind of process. So do I get the same result or the different result?
So if you have a protein, you will definitely do chip-seq for transcription.
Sure.
You cannot do open chromatin assay for protein. Open chromatin assays is irrelevant to transcription factors. Open chromatin assay just says where the DNA is open, where it is not. It doesn't tell you information about the protein form.
will interpret the data of this open chromatin assay?
Right. So I'm coming to the next slide. So
Even
common trend in the
I'm not convinced. If this is protein change you are saying, but you are doing the sequencing at the DNA level, how can you correlate a protein change to the DNA?
Right. So that's why I explained in the earlier slide a protocol about ChIP-seq.
ChIP-seq is only to pull out that part of DNA.
Yes. So the antibody that we use for that specific protein modification, that does this job that you're asking. Okay. So that the antibody pulls that specific protein modification and those DNA fragments, and the DNA fragments get pulled with the antibody, and then the denature it, you get those, and then you get the signal.
How the same thing is correlated with this assay as you have said?
Yes. So like I told you, transcription factor ChIP-seq. In transcription factor ChIP-seq, we use antibody for different proteins which are bound to DNA. Histomodification ChIP-seq, we use antibody for different kinds of histone modification. In open chromatin assay, we don't use any antibody. We just break the DNA and we find where the DNA breaks a lot. So basically, we get more number of fragments where the DNA is breakable.
This is basically a different kind of digestion protocol that we are following. That is a different assay.
Yes. Yes.
But how to decide enzyme concentration for open chromatin assay?
What?
How to decide enzyme concentration? Because at one concentration it could be like more open, at other concentration it could be like less open. So how to decide the enzyme concentration? Like in case of DNA is one, how to decide the enzyme concentration?
Yeah, that is another problem. We use one protocol. So in epigenome, so there are fixed protocols. How much concentration we can use for how many cells. For like suppose we are following DNA's signal. Okay? They are using DNAs to break the DNA, like chromatin DNA. There are fixed amount of, it's already written as a set of rules, and then we use the same all across, like across our study, one study, and then we compare that in this cell, in this region, how much signal we got. So we compare the open chromatin signal using the same assay from continuous cell types, like genes and sample cell types. Right.
So cell concentration, cell type will.
So that is a part of experiment design. Okay. So for some cells, you may need different concentration here. That's a part of experiment design. So before doing ChIP-seq, there is a hard part of optimization. So I have when I started working on epigenome data, initial 2 years, we just spent in optimization of different experiments. What salt concentration and what enzyme concentration, what. So I literally know each and every ChIP-seq. So and why? So that is a experimental design. So once you have optimized your hand, and you start producing the data using the same protocol, then it comes to the data analysis. So, but doing data analysis, there are some rules. Okay? If we have gene bodies, we have promoters, and gene body has exons and introns, and there are regulatory elements called enhancers and repressors. So same regions can work as enhancers or repressors depending upon the transcription factor which is defined. Okay? And then we have insulators. So how to actually distinguish them in the chromatin? So there's some basic rules that people are following is that histone installation, generally H3K27 installation. When we are talking about histone installation, in all over the world nowadays, we just use H3K27 installation because the antibodies are available, but there are different kinds of histone installations. There are already like around 35 different kinds of histone installations. But people use very H3K27. So H3K4ME tri, so basically it's a trimethylation at the residue lysine in H3 histone. That's what. So So basically this nucleosome has H1, H2, H3, H4. These are named nucleosomes. So we get C terminals and N terminals coming around. So at the fourth residue is the lysine, and if a trinucleation attaches there, we call it a restrictive protein. We profile at which genomic region this phenomenon happens using antibody. And these are generally STK4-MET are generally as active open promoters. Then we have enhancers. These both promoters and enhancers can be active or poised or repressed. Okay? So poised region means that it's not repressed, it's not transcribing gene, but it's active. So basically regions which are poised, the genes that are attached to these regions, they are not transcribing, the gene expression is not happening, but they are ready to run. And both regions have H3K4-NE1 histone modification or H2AG, that's a variant of histone modification, histone, there's a variant of histone, H2AX. And they don't have histone acetylation. So the region has no histone acetylation, but it has H3K4-NE1 or H2AZ mutated points. And it's going to be active in the future. Or if you give a stimulation to the cell, these regions will come out faster than the regions which are suppressed. Okay? That's the important point. Then we have in repressed regions, we have different epigenomic signals, like we have methylation in the DNA. DNA itself can have mutation. And wherever the DNA mutation occurs, it causes compression and formation of capsule chromatin. And these regions are repressed, so they are not available for the transcription factors to bind and so the genes are released. Okay? And
Sir, one more question.
yeah.
So which one is better approach, like bisulfite sequencing or this is protein-based DNA methylation sequencing? Which will you prefer?
That depends upon your choice of study and your cost of study.
Which one is
So
first?
if you are doing RRBS, if you have very large cohort, go with RRBS. If you are going deep into finding the real region, you can go sequencing each base pair, bisulfite sequencing. So that depends upon your choice. There are different methods for profiling DNA methylation.
So what I'm trying to understand is how this method is beneficial over disulfide, because disulfide is one of the most used techniques for methylation pattern.
Right. So see, DNA methylation just tells you the methylation of DNA. Okay? That just tells you where the heterochromatin is and where it's not. But from euchromatin, there is heterogeneity. Where the chromatin is open, there is heterogeneity, and these marks of epigenome will tell you exactly if this region is open, what is happening. Is this an enhancer or is it a promoter? Or is it an insulator? So the job of insulator is like if you have one region of DNA which is active, and the proximal region of the DNA is repressed, what will happen? This repressive mass or the transcription factor which cause repression, they try to elongate the repression to the neighboring region. So there, the role of insulator comes. Insulator blocks this transcription, okay, this repression. So this is called insulator. Like in electricity, if you want to stop the transfer of electricity from one wire to another wire. So and CDCF is a protein, and which works as insulator, and it sits with other transmission factors like protein and regulators and all that. So the job of insulator is to block the leakage of compression or activation from one region of DNA to another. So okay. elongating, we still have different epigenomic models. So there are certain histone modifications which help elongation of the gene, like STK79, mononucleation, dinucleation, STK4, nucleation. When they are present in promoter, you can say that yeah, the gene is transcribed, RNA is moving on, because they help the elongation transcription process. And when the gene elongates, when the polymerase, basically there is a RNA polymerase in the promoters, and this polymerase enzyme has different transcription factors. When this polymerase complex moves ahead, it leaves behind another histone modification called STK36 transcription. If you see STK36 transcription, you can say that the gene is transcribing. It's a post RNA transcription. And there are many other dimensions. And so why I'm talking about this, because there are different consortiums across the world. We follow these general rules and they profile these histone modifications, and there are big consortiums across the world. One is called ENCODE. You might be aware of ENCODE. So after human genome sequencing project, this is another big project in like genomics. So what they are doing here, they have different cell types. Some are perpetualized, they don't die, they keep on growing. And some are from, some are basic cell lines from humans. And they have tried to profile the epigenome from these different cell types. So epigenome means they have tried to profile the transcription factor binding sites, they have tried to profile the histone modification, and they have, it's a very big data, okay? And they publish together. And we were part of IHEC. So IHEC is International Human Epigenome Consortium. This consortium is to profile epigenome in primary human cells. Not like perpetualized. They have not been perpetualized. They have not been made artificially. Primary human cells. So we published our some data with IHEC. International Human Epigenome. This is a primary cell from the human brain. And then IHEC has a sister consortium in Europe. Why I'm telling you, because you guys might be looking for jobs, and you can look at these consortiums and the groups involved in these consortiums, and you can look and you can apply for jobs, and also read your papers. We published autism data, autism paper with 40 plus other papers. So we published it together. And then there was Human Epigenome Project. This was before IHEC, but now it has been merged into IHEC. And we have still another consortium that could overwhelm all the other consortiums in coming years. That's the EPIC, the Epigenome of Plant International Consortium. So this sample is easy to get. The only thing is people are trying to optimize different experimental conditions, because plants, tissues are not so easy to handle, like soft animal tissues. So once they have optimized it for different crops or different plants or different cell types, we'll start getting production. So you can village with it. Already input data is already huge. Okay, so I will make it small so all these consortiums, they follow a general rule, and they recommend to use smallest number of histone modifications to profile your requirements. Like they say, okay, you can use STK1 installation for both active enhancers and promoters, then you can just use K1 installation. But what we found is that it's completely mystery. There are 130 histone modifications, 35 histone installations, and we are just using one histone installation, STK histone installation, to represent all histone installations. What about other histone installations? We did a study, and we did machine learning on a set of active enhancers in CD4 T cells. So we did luciferase assay to find which are the active enhancers. So we tested 200 regions in CD4 T cells and tested for the activity as an enhancer. Using luciferase assay. And using those active enhancers and set of negative regions, I did the machine learning, okay, found which is the best feature of which. So we had like 40 chipset data, so almost like 39 histone modification plus all 2 and CDCF plus S2AZ. And what I found that S2PK20 installation is the best marker for active enhancers. It's even better than STK installation. And it is so good, it has its enhancers have the role to regulate the gene from outside the protein, to non-coding regions. And these non-coding regions actually decide which genes will be expressed or non-expressed. So they are generally cell type specific. So what I hypothesize that okay, if H2PK20 is enriched at enhancers, so it will actually mark cell type specific promoters also. And it came out to be true. I discovered this in 2009, and then when I realized this data in 2011, I said, yeah, it's happening. So we took B cell encode data and generated our own data. So every line is a promoter, okay, sitting upstream of the gene in B cell. So we took the signal and we clustered them. So what we find is that, and we have different classes. We have P1, P2, P3, P4, P5, P6, P7. And when we check the absolute expression, we call it FTKM using RNS. So this is absolute expression. We find that P1, P2, P3, and they are highly expressed. We don't see any difference, even though we will see a difference at the level of epigenomic signals from modifications. But when we take cell type specificity of the expression, like if we have a expression of one gene in one cell, we divide it by the median gene expression across many cells, then we get cell type specific expression of the gene. When we see the cell type specific expression of the gene, we see that the promoters which have H2BK20 expression, they have highest cell type specificity. Because H2BK20 installation, sets of enhancers, and so you see enhancers connect to the promoters. So basically, enhancers regulate cell type specific genes. So in genes there are two kinds of genes. One is cell type specific and one is housekeeping. Housekeeping genes are actually non-cell type. So, and this is a very fundamental finding we published in Genomics Research. But seeing this result, you can say, okay, it could be because multiple system modifications sit there. That's why you have more cell type specificity. But it's not like that. We did an independent test. We sorted the promoters according to different epigenomic signals. We took promoters which had, took top 1,000 promoters according to S2BK20 establishment accounts. Similarly, we took top 1,000 promoters having a transcription factor, or you can say cofactor called EP300. And then we took according to STK20 some establishment, which is the most famous of some establishments. What we found that in this independent test, promoters which have H2BK20 installation has highest cell type specificity. There is another enhancer marker called STK4ME1. It also marks cell type specific promoters, but then you see below the line of 0. Some of the promoters are below the line of 0. It means it also marks some of the promoters which are not active. So you can say poised promoters. So we can say that H2BK20 marks cell type specific promoters and enhancers. And we further showed it not only cell types but even cell states.
So, can I ask one more question? So you're showing one of the modifications by using machine learning. So is this the top predicted one, or there are many more modifications which are in this K2 count?
So this was the top predicted not predicted is modification that would mark enhancement. But there were others, there was a close completion. Whenever you do feature selection, it's not that straightforward problem. So in fact, if you could combine 2-3 external modifications together, you could have even better efficiency. But we were looking for just one, so that the biologist can just profile it.
So which technique was used?
Should say.
No, in machine learning, which we have many variants of machine
We
learning.
use logistic regression model.
Sorry.
Logistic regression.
So only logistic regression model, or there are some other things like neural network, random forest? So you
Yeah. No, we use regression models, linear regression models, logistic regression models to find which is the best feature that can give me least error, or least residual error. Can we explain the error? I'm not going. I don't want to go into the details.
how you selected your top hits? Like on the which basis? Is the p-value basis or the frequency basis?
The residual error. When you have regression models, okay, you have set of positives and negatives. Okay, you have 1 and 0. So if you say positive as 1, negative as 0, okay, then you use these features and you use regression models to see whether we can predict. You can come up with some numbers, and then you compare these numbers with the matches of 1 and 0, and then you take a difference. And this difference is the error. We based on the error, the residual error. So S2 becomes 20, S7 gives the least error. Okay. Now, coming to emerging trends in epigenome. So far, I have shown you, the things that have been happening like before 3 years. But what are the recent epigenome trends? People have done whole genome sequencing of 10,000 individuals, and now they are doing more. That's still possible. Why we are doing it? Because we are trying to. There are different purposes. First, one purpose is that we are trying to find the mutations, the genomic variations from different individuals, and trying to correlate that with the gene expression, or we're trying to correlate that with the phenotype. So there is a concept called quantitative trait rules. So what we do is that when we detect a mutation, like we have the genome sequence, every nucleotide is present in 2 forms. So there are 2 copies of DNA in our genome. When they have same allele, we call it homozygous. When they have different alleles, called heterozygous. When they have, so what happens that when we have the genome sequence of the individuals and we have epigenome profiles in one cell of those individuals, one cell type, we can correlate the alleles and particular nucleotides with the signal of the epigenome or the histone acetylation or Roman chromatin signal. For example, this is a part of our autism paper. So at this site, suppose this site is lying at a gene in the intron of the gene. What you see here is that particularly at this nucleotide, we have allele of GG, GC, and GCC in different individuals, and the signal of STK27 acetylation, the CHIP6 signal of STK27 acetylation, is correlated with the allele. Okay, so this is what we call as quantitative trait locus. And it's very costly to sequence. I mean, it was costly like 2 years ago to sequence the whole genome or genotype. What we do, and even if you sequence, you don't get that depth because it's easier to say that okay, we can have $1,000 human genome, but if you just do $1,000 human genome, there will be so much error because you need to have a lot of depth. You need to sequence it 10 times or 40 times more so that you can call exact mutations. So in order to overcome that problem, what we do is that since we have done the ChIP-seq, we have the tag counts or the DNA fragments, small DNA fragments from different regions. We use those sequence of DNA, those DNA fragments to find the mutations. So we use a ChIP-seq signal to find the mutations, as well as we use a ChIP-seq signal to study the regulation. And then we correlate those mutations, allele, with the signal. So it's called HAQTL. You might have heard about GWAS study. So in GWAS study, what we do is that we genotype the individuals, one sample of individuals, and then we say, okay, these many individuals are patient, these many individuals are control, and then we find the odd ratio of that if this individual has this allele, A allele at certain location, then what is the chance that he is more predisposed for disease? So a new concept is coming as HAWAS, histone-wide studies, association studies, it's called HAWAS. So a new term called HAWAS is coming. And soon in coming years, you will see a lot of HAWAS studies. So in HAWAS studies, what we do, we take one cell type from different individuals. So you can't do it because you can't compare like different cell types because epigenome, the assumption is that epigenome is same in the same cell type across different individuals. That's the assumption. But you can have different genomic sequences across different individuals. Okay? And then in between those we try to find the correlation. So you can see here that the peak height across more than 100 samples is actually correlated with the allele at this location. So if at this genomic location, if we have CC, it has lower peak height. If we have GC, heterozygous, we have in the middle, and then if we have GG, we have the highest amount of signal investigated. So HAWAS study is coming. It's coming in the next few years. So DiLaGio. Now, a second emerging trend, that is single cell epigenome. So far what I have been talking about is doing epigenome profiling from 5 million cells or 100,000 cells. But now, as I told you that as we start decreasing the sample amount, we get noise and we lose a lot of information. Because if you see RNA, RNA has multiple copies in cell, but DNA is just one copy. So single cell epigenome is much more challenging than single cell gene expression. So we are not yet there yet, but papers have been published for single cell open chromatin, published in Nature, and single cell DNA methylation, the methods. The single cell ChIP-seq also has come in Nature Biotechnology. So why do we need single cell epigenome, by the way? I mean, come on, we can do it in bulk and all the cells are similar, but it's not true. All the cells are not similar. I have been studying, I had a grant in cancer epigenome. I have been studying single cell data of cancer cells. And my colleague Debarka has been struggling with this data. So we had 900 oral cancer single cell. So we took cancer cells from cancer model of the oral patients. and we have single cell gene expression, and each of these rows and columns shows one cell. And so basically, based on the gene expression, we tried to find the correlation between cells, and based on the correlation, we plotted what are the cell types. So each of these blocks actually shows you one cell type. So we can assume that, okay, this region is, they represent one cell type. But even if you take these cells and you apply drug, what happens? They show heterogeneity in the drug response. So it's very hard to actually, classify the cells or cell response just using gene expression. Because of the concept of the poising, as I told you. So, how to see the poising? So this is what, gene expression tells you the current state of the cell. But epigenome tells you that the past and the future step that is possible for the cell. So if a region is poised and you apply external stimuli to it, that poised region has more chances to be active than the regions which are repressed. Okay. And that you cannot find using gene expression. So whenever there is a dynamism, like response, or immune response, or drug response, single cell epigenome can explain the heterogeneity. So if you take 1 billion cells, okay, they are same cell lines, taken from the same patient, okay, but when you apply drugs, some survive and some do not survive. Why? Because their epigenome was already different, because their origin was different. So in a tissue, we have different kinds of cell types. And these cell types can take cancer state or not. And when they take cancer state, they look like similar, but based on the origin, their epigenome is also already poised for going to different directions. So single cell epigenome is needed to understand the drug response, the responding behaviors, and the heterogeneity in that. And I was almost going to profile the single cell ATAC-seq for cancer cell, but then I came here. But anyway, we will do it. So, because we are so much frustrated in 2 years of study that what the hell is happening? We cannot explain the drug response. They look similar, but why? Because these cells might be having different origin, might be having different poising, different potency. How to find that? You have to go to single cell epigenome. And this is a noisy and hyperdimensional data.
So if I have to compare two components like mutation and epigenome that changes, which one will contribute more to the drug response?
Yeah, that's a very good question. So people are doing this study, and sometimes mutations, sometimes epigenome poison. And we are coming up with a public where we are claiming that, it's the epigenome. We are going to submit in a few months. This is the epigenome that decides the response to the drug. And also, of course, DNA mutation is. But DNA mutations have also had to go through the epigenome. Because epigenome is the main blocking point through gene regulation. Without epigenome, you cannot actually control the gene regulation. Okay, so I would like to acknowledge, my postdoc supervisor, Dr. Chan Pravakar in GIS, the director of GIS, Hakui, and my friend Murali Masafumi, with whom I have 3 publications. He is at Tokuba University, Japan. If you're looking for jobs, bug him. And he's looking for bioinformation because he is sending mouse to space. Because human is poised to invade space. Okay? And So they are doing a study that if human lives in space, what will happen to his cells and muscles? So they are sending mouse to space, bringing them down and trying to study different parts of the mouse, what is the epigenetic. So they have case and control, mouse from space, mouse from Earth. They're trying to find the difference in the epigenome signal, as well as the transcription signal, as well as the DNA methylation. So they have a lot of money in that. Then Nirmala, who has helped in my genome research paper, Sam Benji, with whom we published autism study. Then Postman did the chipstick for the autism study, and then we have Ricardo, who developed this method of H-Avast, HAQTL, and Bing Lane. And then thanks people who have organized this workshop. Ganesh Bagla he took the initiative, and we have the Center for Computational Biology to support us. And Shipra, who is really worried about all small things, very hard. And IT team, whose contribution you can see when you will be going to the lab, who have actually compiled all the programs for you and set up the things. And finally, to my last slide, I started from diving and I'm ending in diving, because it's not a sea of continuous state anymore, it's an ocean. And in this ocean, you are looking for the pulse of regulome. So if you are a diver and you want the most valuable pearl in this ocean, and other divers have already, scratched the surface and taken out whatever was visible directly, what you will do? What you guys going to do? You say, okay, it's already separated, what will you do? Then you have to look beyond your sea, follow the pattern. So the most valuable pearl might be lying there, but the pattern, you have to follow the pattern of the ridges that oyster might be lying. So basically, it's the pattern in the epigenome. So the next challenge is doing machine learning on the patterns. So the question is, can we actually predict the function of the genes just using epigenome and transcriptome data? Because genes, one gene has different functions. There are 30,000 genes, but if they come together, they create billions of functions that we are still unaware of. So, and every gene has gene ontology data set which curates the function of genes, still incomplete. Can we predict the missing gene ontologies of genes just looking at the pattern of epigenome? That's a challenge. That's a human machine learning problem. And can we look at the memory? As I told you, in cancer cells, they have the memory of poisoning to drug response. That you can never find by gene expression. So can we look at the pattern in the epigenome and say, what is the memory of the cell? So what was its precursor cell? Okay? Yeah.
Like finding the most valuable part, it's like a most rarest of the event. So,
It's not most rarest. It's not most rarest, but most valuable. Like, for example, everybody is vaccinated. Okay? Nobody is. The epigenome study for vaccination has not been done. It's not rare. Everybody is vaccinated. How the vaccination goes into the memory of the cells of immune cells, and it sits in it sits in many places. The memory sits at many places. But large part of it is also sitting in the epigenome.
Is it not the adaptive immunity, sir?
What?
You are saying vaccination, is it not the adaptive immunity what you are saying?
Yeah. There, poisoning has a big role. You read a paper in Cell about ligand enhancers. There they've talked about it. They've done it on the immune cells. So, can you find a pattern for the memory? So, people have shown that if you take, when the organism develops, when suppose mouse is born and it grows into adult, if we take the cells of an adult and they do the DNA methylation profiling, the regions which are hypermethylated, regions which are hypomethylated. So they are hypermethylated and hypomethylated. So regions which have methylated means the DNA. I'm talking about DNA methylation. The regions which have low DNA methylation, they are neither active nor poised. What the hell they are doing? And why these regions are hypomethylated? When they look back, and they say that these regions were actually active enhancers in the early stage, in the powerful small, or in the embryonic stage. So it's a memory is residing there. Okay? And in future, those hypomethylated DNA methylation regions can decide whether the cell can become cancerous or not. So new theory is coming in cancer about memory during your embryonic growth. As we already know that children who are born with low birth weight, they are predisposed more towards diabetes and metabolic disorders, obesity. Why? The memory is there. So, and that memory can be at gene sequence level or epigenetic level. Okay, so I'll finish my talk and I will invite, is Dr. Devarka here? I will invite Dr. Devarka for giving his talk. And
So are there any more questions that you are catering to?
Yes, sir.
Yeah.
So can you please tell us the relation between open chromatin assay and FIT? Like how we can use this? Because as you are saying, FIT is based on the antibodies, but open chromatin assay is just the addition of the.
yeah, that's a good question. See, open chromatin tells you where the DNA is available. So it's a different kind of signal. People what they have done is that they have added information of the underlying sequence with the open chromatin to predict whether a protein will bind there, or protein is binding there or not.
So how to identify that what protein is binding there?
Yes, that's another challenge. So people generally use the underlying sequence. So people so researchers are developing methods to predict whether the protein is bound there or not, using the open chromatin signal as well as the motif or the DNA region, the DNA pattern that is favorite for the protein. We call it as motifs for the transcription factors. So they take this motifs signal and then take the DNA signal and then using these both signals they predict whether this protein could be binding. But then there is ambiguity again. Multiple proteins can bind to the same motif. Then you have to also take the information of the gene, of the expression, whether this protein was expressed in that cell line or not. Okay? Got it?
So I can use the data of mass spectrometer proteomics data, then combine with open chromatin assay as you are saying. Like,
Yes, that is possible. But in mass spectrometry, the problem is that you can do it in only one genomic location. It's not genome wide. Huh?
One genomic location. I didn't get you what you said.
So mass spectrometry is for one protein complex. So if you have one protein complex at one location of the genome, you select that region and you do the mass spectrometry and you say, okay, this is the complexes possible. This is the possible complexes here at this genomic location. But here I'm talking about genome wide. For that you will have to go through an exome sequence.
In the autism study, you have postmortem samples. So that there is a study that says that postmortem there is changes in the gene expression.
Right.
So what would be your concern for comparing gene expression as well as methylation, which is the data which you have generated?
Yeah, we did that. It was my part. And I can tell you it was the hardest part of the study. It's very tough. I try to correlate the differential expression with the differential histone modification. Correlation we found with significant p-value is like 30-40, like differential, because RNA has degraded. That's why what I'm saying is that histone modification can give you a better, clearer picture.
So postmortem, you're saying, is that what I understand? Postmortem, RNA will be degraded, so in the sample, we will not be able to capture in the transcriptomic profile, but if we're doing
Yes, I think.
correlation patterns there, then
Yeah.
they are still the same. So we can capture the
That-
biological snapshot in a better way.
Not only that, using epigenome profiling, you can do it for samples which are frozen in Siberia from thousands of years. So that's what people do. People are doing it. So basically, mammals have died long time back, but some pieces of mammals are still frozen in the Siberia. We can do epigenome profiling on that to get picture. Yeah.
What is the sample amount difference between what we use for transcriptomic profiling or
What?
what is the sample amount of samples? How is it different from transcriptomic? For RNA sequencing, we need very little amount of
Right.
sample as compared to does it change with
Yes, yeah, definitely. For ChIP-seq, normal protocol is to use 5 million cells. In RNA-seq, you need less number. And like you can just see the example of single cell expression and single cell epigenome profiling. Single cell expression signal is still much more cleaner if the RNA has not degraded. And single cell epigenome is not, because we don't have that much information coming by people are still optimizing the protocols. So of course, current scenario, at least we need 100,000 cells for epigenome profiling, bulk clean.
and is it that a concern of depth when we while sequencing? What is the concern for the amount of data which has to be generated for it?
Right, same. So for RNA sequencing, at least in bulk, we need almost same number of reads. But if you are doing differential gene expression, you need deeper in RNA sequencing.
One question related to postmortem epigenome studies.
Yeah.
I assume that the protein will be degraded by the time this DNA
See,
expression
the
is
protein
started.
degradation is slower
Okay.
than the RNA degradation.
Yeah.
If the sample has been
Right,
frozen.
right. When you compare RNA, protein is slower.
And also, these samples can be fixed by formaldehyde after taking out. So once they are fixed, they don't degrade so fast.
So you are saying the same technique can be used to, like ChIP-seq, to identify the proteins that have DNA and sequencing, same as the normal sample. Postmortem sample can be sequenced same as the normal sample.
Yeah.
So there are technique called 2C, 3C, 4C and high C for
Right.
3D model of genome.
Right.
So how those technique and this ChIP-seq is like similar?
So, see, ChIP-seq gives you one-dimensional signal, like where this protein is enriched, where this protein modification is there. Chromatin interactions tells you which part of the DNA are interacting closer in 3D. And that's what it is. And chromatin interaction is being combined with ChIP-seq data to actually study regulatory factories that I told you. So basically they found that the genes coming out from one regulatory factory, they have similar level of expression in one cell type. And they might be having shared functions also. Yeah.
So we are saying epigenome is very important part for like pre-programmed or
Yes.
well-state programmed. So
Yeah, so you can go into the future and past of a cell using epigenome.
So these three high C techniques are good. Or this,
Yeah, high C techniques are also there. So see, high C will tell you only one thing: whether they were close together in like. Sometimes it may happen the regions are close together, but they are not active. So you need ChIP-seq signal anyway. ChIP-seq signal will tell you whether that region is poised or active, or was active in future, or was active in past. Okay? And IC will tell you, okay, they are closer. But nowadays, studies are also coming where they are comparing the chromatin-chromatin interaction across patients and control. They are doing it. And so, but overall, what we have seen that 80 to 90% of the interactions, chromatin interactions, are similar across all cell types.
So when we are saying some part is poised, then how we are deciding that it's a poised part based on the that particular modification you
Right. Right. Yeah. Thanks.
Thank you, Rob. Dr. Vimor will be available to you even in the break time as well as later at the time of when we are having hands-on session. Both of them will be available to you. So feel free to ask them questions. I'd like to check out something. How many of you were not formally registered, shortlisted and therefore not registered, and are still here for the lectures? Can you please raise your hands? Okay. So have you guys already given your names at the registration desk? Those who have given, fantastic. Do you have a separate name? Yeah. Those who have not given, please provide your names at the desk so that we have it for our purposes. Secondly, how many of you, those who are not registered and will still be wanting to participate in the laboratory session? Okay. So please ensure that while you are giving the name, or if you are not, if you already given the name, even then you inform these to Shipra for the registration purpose, so that we are having enough seats for at the time of training session. Okay. We will have a break now. So we have a tea break and snacks break, and we expect to be back exactly in half an hour. So you get 11:20 is when we should be coming back. Okay, so let's make it 11:25. By that time you should be back here, so that by 11:50 latest we should be starting the session. Okay, the registrants are available in the last chamber. They can go ask.